So good evening, everyone. I'm Prachi Pasalkar. I'm Program Manager at Capacity Building. And we are all here for a very interesting talk by Dr. Sudha Rajamani. And she's going to talk about astrobiological narrative of life's origin. So it's a very interesting talk. So Dr. Sudha Rajamani is an associate professor at Indian Institute of Science, Education and Research, IISER Pune. And her basic or her research area is into astrobiology, and what she's going to also talk about to us today. She also have interest in astrophysics and the astrophysics angle to biology and how life is originated and all of that. So we are going to have a very interesting session today by Dr. Sudha. Over to you, Sudha.
Thank you, Prachi. I'm going to start sharing my screen. Can you see it?
Yes, we can.
Yeah, we can see it.
Yeah, okay, awesome. So hello, everyone. Thank you, PKC team, for giving me this opportunity to connect with a wide range of audience via this talk. To everybody that has joined, my name is Sudha Rajamani. I'm currently an associate professor at the Indian Institute of Science, Education and Research in Pune. I'm a biochemist by training and an astrobiologist by profession. And today I'm going to use this opportunity to give you a flavor of the kind of theories and concepts that exist in the field that try to hypothesize how life would have emerged on the early Earth. Fundamentally, the understanding is going to help us try and appreciate how the transition from chemistry to biology might have happened on the early Earth. And what do I mean by that? Okay, before I start off, I just have to tell you that my already really fun slides were made even more funner by a team at Scientifically Speaking in Ashoka University. And thanks to them for helping me put together this really sort of fun PPT. Okay, this is basically pertaining to how we think life would have originated on the early Earth. And nothing would be possible in research without the contributions of a wide variety of participants. And a lot of them are actually very early stage career. They are very early stage researchers and hope to make science their careers at some point in life. And they range all the way from undergrads to postdoctoral fellows. So I've been very lucky to have a whole bunch of really fun people go through my lab over the last 11 years. And towards the end of the talk, I'll also give you a flavor of how, in the context that I'm going to set up, which allows you to appreciate this transition from non-life to life on the early Earth and how we think that might have happened and where it might have happened. Within that broader sort of structure, how can we systematically pose questions and address these questions is something that I want to sort of leave you a flavor of by the end of the talk. Okay, so yes, let's start off. What do I mean by transition from non-life to life? If you kind of look at biology, biology is populated by molecules that show up at the bottom of the screen, right? Let me start my pointer. That will help us a little bit better. Yeah, can you see that? Yeah, so there are a whole bunch of molecules here, some monomers and some oligomers. The idea is to say that these are complex enough that they are made up of a whole bunch of atoms, and they come together. These monomers come together to make much larger polymers, which essentially evolve lifelike behaviors that allow us to think about what kind of functions might have been important for the emergence of life, what kind of molecular structures do we have to think about when we think about the emergence of biology from chemistry. But zoom into these molecules and what do you see? You're now in the realm of chemistry. You're now in the realm of smaller molecules. But again, it's not all small, but I'll get to that in a second. But this particular chemistry that I'm going to be alluding to a little bit in today's talk is not just that happened on terra firma or on our planet. It's also something that has happened or continues to happen in the interstellar medium. Yeah, we're talking about spaces far out in between the stars. And a lot of interesting prebiotic chemistry has been demonstrated to happen in these parts of the cosmos, both using sort of experimental verification protocols that we have done in our labs, and also by looking at them using extremely high resolution tools, spectroscopic tools that tell us the kind of molecules that are present in these regions of the cosmos. And on the right side here is basically a very complex molecule called the buckyballs that has been discovered in the interstellar medium, and it is made up of up to C60 carbon atoms. So they're not really very small, but suffice to say that molecules in the chemical realm are, relatively speaking, smaller than what you find in the biological realm, where we are predominantly talking about much larger polymers that end up evolving emergent properties, which you then cannot attribute back to the single sort of monomers or the smaller molecular inorganic molecules that actually exist in the chemical realm. And if you zoom into those particular molecules, you further realize that they are literally what results from coming together of fundamental atoms which exist in the physical space. So this beautiful realm that exists between physics, chemistry, and biology allows us to systematically think about a sort of a hypothesis that allows us to posit the kind of niches and the kind of environments and the kind of processes that might have allowed for these transitions to take place. In this talk today, my main focus is going to be about how we think the transition from the chemical space to the biological space might have happened. So when we talk about these things, first we talk about the resource material. So where did the fundamental molecules come from, right? The first biomolecules on Earth. So depending upon the setting, it could have happened in two different ways. One is because of all the very interesting chemistry that can happen very much on our terrestrial planet, on our Earth. And another thing, another mode of delivery is via exogenous delivery, where molecules that form in the interstellar medium get entrapped into cosmic bodies that then eventually travel to the Earth in the form of meteorites and comets and bombard Earth even to this day and bring along very interesting organic matter with them. And I'm I'll get to it later on, but some of my colleagues in the field have drilled into the core of some of these meteorites and have shown that they actually have a whole suite of biologically relevant monomeric molecules. For example, they have a whole bunch of amino acids, some of which is not used in life also. And that is an important point to remember, because then the question is, why is it that we see the molecules that we see today in biology, while the repertoire of molecules or the prebiotic soup of molecules that was available to us was much, much, much larger on the early Earth. So this is just to give you a very, very, very sort of quick flavor of the various kinds of molecules that we think might have been present in these prebiotic soup, in these volcanic geothermal pools, for example, which is shown on this right panel here. And this pool of very interesting molecules is thought to have come together from much more complex molecules. So you go from an inorganic molecular space to an organic molecular space where you have sort of interesting prebiotic reactions forming the fundamental monomeric molecules that are relevant to biochemistry. And these molecules are thought to have come together and form much more complex molecules, which then interacted with, and these biomolecules that formed interacted amongst each other to form one of the most important entities that is present in all forms of life, whether it be Nelson Mandela, whether it be a bacteria, a catfish, or a giant redwood, or a mushroom, or even T-Rex. This is basically the least common denominator of all life, and we call them cells. So one experimental paradigm that you can think about when you're talking about the transition from chemistry to biology is to think about how the interesting prebiotic chemistry that happened in the prebiotic soup that led to the formation of relevant biologically interesting molecules allowed for how did the niches and the environments allow for the interactions that eventually led to the formation of the oldest cells on Earth. And these cells, please understand, are not as complex as the cells that are present in your body or some of the earliest forms of life too. These are much, much more simplistic, and I'll give you a sense of how we think about them. So these are called protocells, and they are thought to have predated LUCA. Here you're seeing an ultra simplistic rendition of the tree of life, which at the base shows that there probably was an entity called LUCA, or the last universal common ancestor, from which all the three domains of life as we know is thought to have come about. This is sort of the understanding that has been present for the longest time, but in the last couple of decades, it's getting refined. And we think the more realistic picture is like this, yeah? Way before LUCA, there probably was a common ancestral community of primitive cells. They're not primitive by any stretch of imagination. And I hope by the end of this talk, you will have an appreciation of how complex even these most simplest of entities are. And these ancestral community of cells would have been shaped by a lot of environmental parameters that would have helped for the survival of some, and some of them would have got lost out in this race, eventually leading to this bottleneck point where you would have had LUCA-like organisms, which then eventually led to the formation of the 3 domains of life. So this primitive cell, which I don't want to call primitive anymore, I'll take that word back, primordial cell or the protocell is a minimal sort of living system, which is thought to have been made of a membrane boundary, just like in modern cells, you have a phospholipid bilayer. It's thought of to be made of a bilayer of a membrane, which is composed of much more simplistic molecules that have been much more readily available on the early Earth. And this particular entity, if you cut it open, you can see that it encapsulates some genetic moiety, some genetic entity, which is capable of encoding information and allowing for it to propagate, and a minimal network, metabolic network, that allows it to stay out of equilibrium. All of life processes are out of equilibrium processes, which is why it's even possible for life to exist. So this is sort of the sort of cartoon notion of how this protocell might have looked. We still do not know what are the exact fundamental components of this entity, but suffice to say, a membrane boundary, encapsulated genetic material, and a minimal metabolic network that would have allowed for some form of proto-metabolism to occur that allowed it to sustain itself, grow and replicate, and sort of propagate itself. So what I'd like to start off by saying is that since the field of prebiotic chemistry got formally sort of formally started in the 1950s, early 1950s, we are basically able to gather a lot of evidence from what experiments in prebiotic chemistry are telling us, what geology of the early Earth, which we can understand both using computational means and by actually doing some fundamental experiments. What is that telling us about what, how the geological conditions might have been on the early Earth, and what some of the most earliest forms of lives, like cyanobacteria, the biology of these cyanobacteria, what do they reveal about some of the first forms of life that might have come about? So by using inputs from all of these areas and some more, we are finally kind of trying to put together this puzzle of how this transition from non-life to life might have happened. And finally, after a century of very interesting work, the story is beginning to kind of come into view, and we're better able to appreciate the various sort of substructures of this puzzle sort of coming together. And I will talk about some of this as we go along. Very importantly, astrobiology as a field is extremely interdisciplinary, right? You could be a person who's trained in biology like I am and be asking questions in this area, or you could be a person who could be trained in a whole bunch of other sciences like paleontology, chemistry, ecology, astronomy, physics, mathematics. I didn't put it there, but math underlies a lot of these sciences. You could be a person who's trained in any of these various areas and still be pursuing research projects which has fundamental implications for understanding how life emerged and how it evolved and propagated, and is there life elsewhere in the universe. Astrobiology encompasses the study of all of this. And in our lab, we basically are very interested in understanding how do you go from this prebiotic soup of molecules. First of all, what might this landscape look like? We still do not have a true idea of how complex this prebiotic soup might have been. And from that soup, how did you form the oldest of protocells, which eventually is thought to have evolved into the most simplest forms of life called prokaryotes, which then eventually led to the evolution of eukaryotes, which is you and I. And the scientific areas which we in our lab sort of touch upon and use tools from to address the questions that we are interested in our lab encompasses a whole bunch of sort of scientific disciplines, as I've mentioned here below. And we also extensively interact with people who work not in laboratories, but on computers, or they literally use pen and paper, and they do modeling, and they have a very theoretical approach to understanding these questions. And we collaborate with them to sort of also bring in a much more holistic picture about trying to gain a true understanding of this whole process. But when did all of this begin? How far back does the story go? And here is a timeline that gives you a flavor of that. You're looking at a timeline which is in billions of years ago, and roughly about 3.8 to 4.2 billion years ago is when this very interesting grand drama of origin of biochemically relevant monomers and their transition to a minimal informational world to have happened is thought to have happened. How did this happen is something I'll touch upon, but this is the period in which it's thought to have happened. And subsequent to that, you had certain evolutionary processes that then allowed for life as we see today, where you have 4 rock star biomolecules, right? Nucleic acids, proteins, lipids, and carbohydrates that facilitate all of the amazing biochemistry that happens in our cells that allow us to have this conversation today. But prior to that, it is thought that there was an early period during the emergence and early evolution of life when predominantly one molecule did most of the jobs. And this molecule is the RNA molecule. And this particular hypothesis is called the RNA world hypothesis, which posits that given interesting prebiotic chemistry that led to the formation of RNA monomers or RNA monomer-like molecules, you could have had processes and environments that would have allowed for the polymerization of these molecules via certain pre-RNA world molecules into the RNA world molecules, where both the function, both the information encoding is in the RNA molecule, and RNA molecules themselves facilitate enzymatic catalysis. And these molecules are called ribozymes. So now what this does is it reduces our problem of trying to understand this transition from non-life to life or chemistry to biology, of trying to understand how do you go from the prebiotic soup to a space where you had an RNA world emerge, and how molecules of this RNA world interacted with the other biomolecules, either in the monomeric space or the polymeric space, to eventually result in early cellular life. So the environments where these things might have, very interesting things might have happened. So depending upon whom you talk to in the field, there are 2 prominent environments that come to the fore. We simulate terrestrial geothermal pool-like conditions in the lab to carry out some of our experiments. However, you also have these volcanic environments present at the base of the ocean, and these are called vents. And here you're seeing a hydrothermal vent, which is a black smoker. It's called a black smoker because the gases that come out of it essentially impart a black color to it. It's because of the aqueous, it's because of the chemistry that happens because of this aqueous water interaction with the minerals present in the environment. Both these environments are very interesting because they provide very interesting chemical and thermal fluxes that essentially allow for interesting uphill reactions to happen, far from equilibrium reactions to happen. What do I mean? Let's take the RNA world hypothesis as an example. RNA molecules are long polymers, just like your DNA molecules, like your protein molecules, like your carbohydrate molecules. They're all long polymers where individual monomers come together, form bonds between themselves, and form these long polymers. So this transition from the monomeric to the polymeric space is essentially thought to have imparted some special functions to these very long polymers, which is why life is thought to have been possible on Earth. Now, going from this monomeric space to the polymeric space is completely non-trivial because it's a thermodynamically uphill task. If you want to bring, for example, 2 RNA monomers together to form a dimer, you need to knock a water molecule off, which results in a phosphodiester linkage. And this seemingly small reaction, a simple reaction, is not at all simple. In your cells and my cells, it's facilitated, orchestrated by a whole suite of enzymes. It's an energetically uphill process, so ATP is used, and all of this drama happens, right? But back on the early Earth, you have a chicken and egg problem. Did the nucleic acids come first, which encoded the information from which you could form proteins? Or did the proteins come first, which are very important for making long chains of nucleic acids? Now that's a problem. That's called a chicken and egg paradox. And this is why this RNA world hypothesis provides a beautiful respite, because you can now think about just chemical ways of activating a RNA monomer, and they can very readily string into polymers under relevant geothermal conditions. And I will show you how that is possible. So just to kind of reiterate, we in the lab try to simulate these terrestrial geothermal conditions to understand how these processes happened. And the original stage was set way back, way before 1953, right? Because of this beautiful theory that was postulated by two gentlemen, one a Russian and one an American who has an Indian connection. This is Oparin and this is Haldane. Fundamentally, both of them posited that if you have the right ingredients in the primordial early ocean, like ammonia, carbon dioxide, methylene, hydrogen, under those very reducing conditions, if there was a very high energy source like lightning, ultraviolet radiation, you can spontaneously have transition from this inorganic molecular space to the organic molecular space, here represented by a string of amino acids. So what they basically posited is that in this primordial soup, given the right conditions of temperature and if it had the right kind of ingredients, which is, I already have told you how we think the fundamental ingredients might have been present in these environments, you can essentially chemically think about how, just because of fundamental chemical principles, physicochemical principles, you can think about how the transition from inorganic space to an organic molecular space might have potentially happened. So all theory is theory till you systematically demonstrate it using an experiment. And that is where the next important big step in the field came about. It happened in the exact same year as the year when DNA structure was described, and that is 1953. In 1953, this is Stanley Miller, he joined Harold Urey to actually demonstrate experimentally the Oparin-Haldane theory. This is the cartoon representation of the setup that Stanley Miller used. And here is Stanley Miller sitting with his original setup in which he undertook this experiment. This particular round section of the setup is where they simulated the primordial ocean, which had methane. Sorry about some of these words looking a little wonky, but basically they had methane, water, ammonia, and they kind of heated it up. And why is heat important? Heat is important to facilitate condensation. And condensation is a very dehydration, removal of water molecule is a very important step in several chemical reactions. And it would have been very readily possible on the early Earth because of all those high energy sources I was talking to you about, like lightning. And the fact that your early Earth was not an ambient, gorgeous place like it is today. It's thought to have been very, very hot. And depending on whom you talk to, it would have been a balmy 60 degrees C. And back then 60 degrees C was balmy. It would have been either anywhere from 60 degrees C to 100 degrees C, depending upon some of the geologists that you speak to, because there are different indicators for this, and we are still not sure about what exactly the temperatures were, but collectively there is understanding that they were very high, the temperatures were very high. So and at these high temperature conditions, you can very readily have all of the water sort of condense out, and you can now have the molecules go along with the water vapor. And in this particular space, you basically have a tungsten electrode, which is simulating your lightning. So this is the spark discharge, which they applied to this set of chemical molecules, which then undergo very, very interesting reactions. And the fumes that come out can be passed through this condenser. This is actually a pipe, and surrounding the pipe, there is a setup which allows you to push in cold water, which allows the gases now to condense into liquid again. And basically, this cycle can keep going on and on and on. What Stanley Miller went on to do is, in frequent intervals, he was collecting eluates from this position to systematically analyze them, and using extremely simple but elegant techniques, including paper chromatography and thin layer chromatography, he went on to show that from a mixture of these inorganic molecules, you can form a whole bunch of these very interesting biologically relevant monomers. I'm showing you amino acids here like glycine, alanine, glutamic acid, some of the other fundamental hydroxy acids like formic acid, glycolic acid, acetic acid, and also molecules like urea were very readily formed. This was an experiment done in 1953. Since then, there have been several groups in the field that have tried to recapitulate or redo this experiment to see if it works exactly the same way if you repeat it using the same parameters, or if you change the parameters, does this product distribution change? So I will just talk about one of these experiments, which was conducted in early 2000s by Miller's first student, Jeff Barra, and one of his last few students, Jim Cleaves, who's a dear friend. They went on to work with the exact same setup, old setup, and they redid this experiment to show that, yes, indeed, you're forming not just this molecules, a whole bunch of other molecules, including a whole suite of other amino acids, which they could detect even though they were present in extremely small amounts using much higher end instrumentation that exists today, something like ion coupled mass spectrometry. It's a very high end mass spectrometry method that allows you to detect molecules even if they're present in parts per billion amounts, right? So they went on to show that indeed you actually can very readily go from this space to this space, and not just it's not just that you have 10 to 12 interesting molecules form, you have a whole suite of molecules very relevant to biochemistry that can readily form under these reactions. So there is this model that we work with, which allows us to think about the steps in a little more systematic manner, which basically goes this way. If in one of these very interesting niche, here you're seeing a tidal zone, you remember how there are these tidal zones. When there is high tide, there's a lot of water. When there's low tide, there's no water. It's a lot like a volcanic geothermal system where during the hot day, during the day, water evaporates out, and during night, water condenses. So there are these very interesting bed-rise cycle events that are naturally facilitated by geochemical processes, including day-night cycles, seasonal variation, so on and so forth. So if you had the monomers that form the protocell present in these scenario, let me for a second just talk about the membrane molecule. The membrane molecule in our cell, the main membrane component is a phospholipid. A phospholipid has one water-loving head group and two water-hating chains. It's a diacyl lipid. But very early on, it is thought that you didn't have diacyl lipids, but you had monoacyl lipids like fatty acids, which is one of the important fundamental ingredients of our phospholipid. It's thought that these might have very readily formed these higher ordered cell-like structures, and they very much do. If you had them in aqueous conditions, because the water-hating groups hate to not basically despise interacting with water, they kind of clump together to form these higher order structures called come together to form higher order structures like micelles, bilayers, and vesicles. And in the same vicinity, if you had monomers of informational or catalytic polymers, like that of RNA or like that of small peptides, amino acids, right? They are the fundamental monomers of peptides. If you had them in the vicinity, multiple cycles of dehydration and rehydration in principle could allow for the growth of polymeric entities from these monomeric entities. So here you're seeing this vesicle, which is formed here in the aqueous conditions. When you take out water, it dehydrates and flops into this multilamellar structure. And this is a real image that you're seeing, a freeze fracture microscopy image of one of these structures. And if you did have polar molecules in the vicinity, they very readily now are constrained in a two-dimensional space, which facilitates greater interactions. And because you're pulling out water, you facilitate condensation reactions, which form polymers. And in a subsequent rehydration step, you can very readily have these multilamellar structures bleb out and give you vesicular entities again. And now the cool thing is these vesicular entities have entrapped the growing polymer. Like you remember I told you about the illustration of a protocell. You have a membrane. It has an entrapped genetic material. In this case, these strings of red polymers are thought to be RNA. Yeah? And multiple such rounds of dehydration and rehydration are thought to have also facilitated some kinds of minimal selection where some of the fitter molecules might have stayed around while the ones who couldn't stay around just broke up and fell apart. Yeah. So some small early version of selection is thought to have also driven evolution in these spaces. And these multiple cycles is thought to have set the stage for the emergence of protocellular life. Here in this inset here, quickly, this is a real microscopy image again. And this is fatty acid and a derivative of fatty acid called fatty alcohol taken in 1:1 ratios and mix, they readily form lovely, beautiful tubular and spherical vesicles. And here they have been, we basically added a dye which can get encapsulated into its lumen, which is why it is fluorescing green, showing you, giving you the idea of contours of how these look, right? So in all these years since 1953, I'm very happy to share that we have been able to demonstrate, people in the field, colleagues, dear colleagues in the field, have been able to demonstrate that you can go from a whole bunch of these simple monomers to amino acid precursors that are important for forming proteins, nucleoside precursors important for forming DNA, RNA, and lipid precursors, which are important for forming your membrane structures. So if this recipe for life itself was indeed plausible chemically, then the next question is, how do you go from this monomeric space to a polymeric space? So that is where we think this is how, these are the various steps that might have been involved once interesting prebiotic chemistry led to the formation of building blocks. In the absence of enzymes, you need to think about ways in which non-enzymatically you can polymerize monomers to form interesting oligomers like RNA. And I hopefully have convinced you that you can still do that by invoking interesting, relevant geochemical conditions. And if you did have polymers come about in this RNA world space, some of which could evolve some function, because RNA has this wonderful capacity of falling back on itself and folding up and forming pockets just like protein enzymes where interesting catalysis can be facilitated. If some of them did indeed have this function, one of the important steps is to think about, one of the important things to think about is, how did the information of that functional polymer get reasonably faithfully transferred even in the absence of enzymes? And then I just put this on the side because we are still not sure when all of these events might have happened, but formation and the higher order assembly of primordial membranes or early membranes would have been very important so that the crosstalk between the two of them can be facilitated. And there was this altogether another drama happening in the proto-metabolism realm where some of the oldest forms of enzymes or minimal enzymes were coming about, and all these molecules interacted to eventually lead to the formation of protocells. So what do we do in our lab? We work on different aspects, and I'm sorry about that. We work on different aspects and we collaborate with a whole bunch of people that I've mentioned here. And we are interested in understanding how informational molecules and even catalytic molecules, though it's not mentioned here, came about under some of these conditions. What is the protocell landscape? What is the early lipidic landscape that led to the formation of the first protocells? What might have their compositions been? What are the kind of error rates associated with information transfer in the absence of enzymes? How fast are these processes? What is the fidelity of this process? How did it allow for generation of complexity even when there were no enzymes present? That is something we like to learn about. We're very interested in thinking about how some of the first catalysis happened. And a lot of it is thought to have been predominantly mediated by metals. This is inorganic catalysis. And a lot of interesting mineral catalyzed reactions are also possible, and that is also thought to have happened. And when there was interaction between these metals and minimal scaffolds and small peptides, you can sort of envisage a evolutionary pathway of how you go from simple metal-mediated catalysis to a space where you can have catalysis now driven by minimal enzymes. And all of the work that we do in our lab has fundamental implications for thinking about habitability. When we say habitability, what do I mean by that? Basically, it's about looking at other planets, not just in our solar system, but even outside of our solar system that have that probably had a similar geological history, is a rocky planet, and has at some point harbored water or has water. If you find these extraterrestrial bodies, can we think about manners and ways in which we can experimentally or theoretically, systematically think about how these planets might have been habitable, or can they become habitable? Can they be made habitable? So on and so forth. There are many implications actually. And in order to understand how all of our processes actually pan out in a realistic environment, not just in extremely pure controlled environments of the laboratory. We also step out of the lab and we do studies in analog environments. There is one such Mars analog in our country, which is Ladakh. Ladakh has a lot of interesting terrain features that makes it a Mars analog. So we basically go out there, we do some experiments out there, we bring back a lot of samples from there to complexify our starting reaction mixtures and make them more realistic to better understand if the fundamental prebiotic reactions that we're interested in, are they truly going to pan out in the same way when you step out and do them under realistic conditions? Because those will have direct implications for actually thinking about creating an instrument that you want to put on a rover and send out to, for example, actually Mars Perseverance rover is precisely doing that right now, to try and understand if there is if there are molecules out there that are interesting from a biochemistry perspective, based on how we understand life, so on and so forth. Yeah. What time are we at now? 4:41. So do I have some time to talk about some of my own projects? Like a couple?
Yes, Sudha. Yes, you can.
Okay. So in one of the very first projects, my very first student, whom I'm super proud of, Chaitanya Mungi, who's right now a science outreach and science pedagogy person himself, he basically wanted to understand if you could form pre-RNA world monomers from the prebiotic soup. Because due to lack of time, I'm not going to go into details, but going directly from the prebiotic soup to an RNA world space is completely non-trivial. What we can readily form are basically oligomers with abasic sites. And these are oligomers that do not have the informational moiety intact at every single position. What is more readily plausible is to form these sugar phosphate backbones. Here I'm showing you ribose molecules, which are interlinked by phosphate. What Chaitanya showed is you can much more readily form these sugar phosphate backbones. So now we are thinking about another way of forming informational polymers, where if you had the sugar phosphate backbones and if you had a whole bunch of monomers, not just A, U, G, C, and T, which is what we see in biology today. These things are probably a product of billions of years of evolution. Yeah, they probably were not the rock star informational molecules back on the early Earth, because you need to think about stability of molecules under harsh conditions. How can they readily react to form interesting oligomers? There are a lot of different parameters that come into play. So he started out a project where we have since then been trying to understand how do you go from sugar phosphate backbones to pre-RNA world molecules? What are the various informational polymers that can readily form under those conditions, which are not necessarily made of only the extant bases that you and I read in our textbooks. And he also actually showed that if you take the ribose sugar with the phosphate attached to it, you can actually condense some of this alternate bases, not AUGCT, but alternate bases, which look a lot like your modern bases, but they have the exocyclic atoms that can interact with each other placed a little differently. And he showed that you can very readily form a prebiotic nucleotide. In this case, this is a cousin of uracil called barbituric acid. And he went on to show that under early Earth conditions of wet-dry cycles in volcanic geothermal pools, you can very readily form this molecule, while you cannot form your AMP or your GMP or your CMP or your UMP readily at all. And this basically is a study that we sort of worked our way because of some of the reaction products that we were seeing. But there is this hypothetical evolutionary lineage of nucleic acids that was described in the field by a very dear friend and colleague of ours, Nick Hudd, who actually has discussed about these sugar phosphate oligomers, as you see here, right? So Chaitanya went on to show this, and since then we have also gone to form certain kinds of pre-RNA world monomers in the lab. And he was ably assisted by Anupam, the postdoc in my lab, who's a synthetic organic chemist. And Shikha also worked on trying to understand how a certain kind of RNA monomer. RNA monomers are of a few different kinds, and I'm not going to go into details. Depending on which monomer you start from, some of the product spectrum will vary. And so these are systematic characterizations that have been done predominantly by Chaitanya and Shikha. In a second project, Nirja, my second student, another stellar student, set out to understand what the role of prebiotic heterogeneity of the soup means for some of the templated replication reactions that can happen in them. When I say templated replication, the assumption is that you actually would have had a scenario where there was already an RNA molecule that could form a template, and there were shorter primer molecules that are present in the prebiotic soup. Some of them which can complementary base pair with the template here can allow for formation of longer new strands if you give them enough chance to sort of go through the cycle. But when we test some of these reactions in the lab to understand its kinetics, its fidelity, we do them under very, very pure conditions. So of course you get some numbers, and of course you interpret them in a certain manner. But if you now do these reactions under much more realistic conditions, it was very unclear in the field what exactly might happen. And she went on to show that in the presence of prebiotically relevant co-solutes, some random, some that directly are participating in the reaction or are competing with the reaction, she went on to try and understand what happens to some of the rates of reaction that happen in these more realistic conditions. And she showed that you have much higher mutation rates. It's good and bad, and I can talk about this later in discussion. Gauri went on to take over this project and has done some very cool work since then. Kshitij here, who's one of the undergrad students who's currently doing his PhD in Germany, went on to understand the crosstalk between the lipid membrane and the monomers of RNA. What does that mean for the properties of the membrane itself? Because every time we talk about crosstalk of molecules, emergence of protocells, some of the fundamental things that you need to understand is what the presence of one means for the other and vice versa. So those are other kinds of studies that also are something that is happening in the lab. I will skip this. In a third big focus in the lab, we are very interested in understanding the protocellular landscape of the early Earth. A lot of the initial reactions in this field were done using simple molecules called fatty acids. Fatty acids, like I said, have a head group and a tail. Here is a fatty acid, sorry, it's really small, but you can see that here. A head group containing carboxylic acid and a tail, aliphatic tail of hydrocarbon. So fatty acids and some fatty acid derivatives are the molecules that have been predominantly studied in the context of vesicle formation, how do vesicles affect, how basically growth and division could happen in vesicles, so on and so forth. But Sushaman joined and he started out a project where he wanted to sort of take this approach in a much more systematic, take this approach further in a much more systematic manner to better understand what this protocellular landscape might mean, because physicochemical parameters like pH, presence of divalent cations of metals, right? And presence of high temperatures and certain regimes with certain kinds of chemical moieties, all of them would have impacted which sets of monoacyl fatty acids might have much more readily assembled into vesicles than others. And what he went on to show with his work, along with able assistance from Shika, is that those which had more compositional heterogeneity, those which were made up of more components, more kinds of molecules, had a selective advantage to survive multiple of these selection pressures. I'm going to leave it at that because it's a very complicated project with multiple sort of directions. In more recent work that's been led by Shoradeep, and Shoradeep is a grad student and Sahil another undergrad student, we want to try and understand how do you go from simple prebiotically plausible single chain amphiphiles, how do you go via the primordial compartments to compartments which were more complex that eventually might have led to the first functional protocells on the early Earth. Again, there's a systematic way of thinking about it. And these two people are trying to understand the various steps involved in this process by looking at this problem in different manners. Like, for example, Shoradeep is very interested in understanding how the composition of the compartment will impinge on their capacity to grow and divide. Finally, in the last project, which was led by Manish and is now being taken forward by Anupam in very interesting ways, basically, we looked at the crosstalk between lipidic moieties, which form the membrane, and peptide synthesis, early peptide synthesis. So the crosstalk between amino acids, peptides, and membranes. And what Manish went on to show is that when you start looking at prebiotic reactions in an unbiased manner, you can actually discover new molecules which have never been seen before in these reactions or were ignored because we are biased by what we understand about biochemistry. So we wear those glasses and we don't look at other kinds of things that exist in the reaction milieu. He went on to show that in a reaction where you put put amino acids and phospholipids through wet-dry cycles, you can actually form a new class of molecules called N-acyl amino acids. These are not new to biochemistry at all. It exists in your cells and my cells. They are very important signaling molecules. This particular experiment demonstrated for the first time that they can readily form under prebiotic conditions, and they have fundamental implications for understanding this prebiotic amphiphilic landscape, because it is just not fatty acids and its derivatives. It would have been a whole plethora of amphiphilic molecules, and what they might be is something that we are trying to systematically catalog. And in a final project, like I said, we're very interested in understanding the oldest enzymes that might have facilitated catalysis. How then did you go from simple hydrated metal ions to actual metal complexes? What kind of scaffolds might have been advantageous? For example, a lot of enzymes today, you will see porphyrin as an important scaffold. Is there an intrinsic advantage that porphyrin got selected over other scaffolds that might have been present? We do not know. And eventually, we are hoping to sort of tie up this loop to basically understand the whole steps involved, all the steps involved. Hydrated metal ions, their interaction with metal complexes to form metallated ligand complexes. And from there, how did they go to minimal enzymes and finally evolve into modern enzymes? And this is something that Shikha started, and Anurag, another undergrad, who very ably took it forward. And finally, like I said, all our work has implications for habitability. And here is the example of an experiment that is currently ongoing, right? So here is your Perseverance rover. It's rigged with a whole bunch of extremely interesting gadgets, which is going to allow us to understand a whole bunch of things. What is the elemental composition of the Martian surface? And what are the kind of materials that are present there? What is the mineralogy on Mars like? Yeah, the SuperCam, for example, is going to be able to directly image the minerals, and not just that, it also has a provision to figure out the chemical composition of these minerals and rocks and regoliths. So all of the fundamental science that I talked to you about and gave you just a small flavor of, because this is a really, really wide, vast area of research, has direct implications for actually sending something like this out to another planet, yeah, whose geological conditions, early geological history was all completely probably different, and to sort of try and see if Mars did harbor life way back in time. Mars is tectonically dead now. It does no plate tectonics activity now. But back then, very early on, when it was possible, there's a notion that it might have harbored life. So we're trying to understand what kind of molecules might be present there that still give us some glimpses of what might have been several billion years ago. So with that, I'll say thank you for your time. Here are snapshots from the fun stuff we do when we get together and not do research. And I am happy to take any questions that you might have.
Thank you so much, Dr. Sudha. It was really interesting talk. It was cool. As the name of your lab, it was really cool talk. People already know that, Sudha's lab has the name COOL, which is chemical origins of life. And yes, it was really interesting to know about our origins. Yes.
Thank you.
I will now hand it over to Gayatri for questions. We have quite a few questions in the chat box.
Sorry, before I forget, a whole plethora of bodies, funding bodies have funded our research. This includes funding from our own Department of Biotech and Department of Science and Technology. In addition to that, we also get some small funds from international bodies that facilitate collaborative work. And thanks to IISER Pune for believing in me and my group and constantly supporting us. Thank you.
Prachi, before you go to the questions, I'll just say a few things.
Yes, sir.
Yeah. So, Dr. Sudha, thank you very much for a really excellent lecture.
Thank you.
And, it was very, very nice and very detailed. But what was very impressive was you're involving so many young people in it. And there are not too many people in astrobiology in India, really. I don't suppose there's very many people apart from you. And the fact that you are involving so many young people means that there's every possibility that the science will grow here. because they will get jobs in other places, and that is the way it should grow, because this is a science for the future, and we really must invest in it. And
Absolutely.
it will very nicely complement the work on exoplanets which is starting. But
Yes.
I don't think that they have so many people working in such a focused fashion on it yet. But hopefully that day will come. And the fact that you are funded also means that you are here. I mean, this work is here to stay.
Thank
So
you.
I should really thank you and congratulate you.
That thankfully there are a few other institutes in the country. It's not a big list, but nevertheless, there are still a bunch of institutes in the country, in other parts of the country other than IISER Pune, where aspects of astrobiological research are going on. Like I told you, it's a very vast area. This umbrella, the umbrella of astrobiology feeds off of lots of other areas
Yes.
of sciences. So there's
Yeah.
work happening in Indian Institute of Astrophysics, and Amity University has a Center of Excellence in Astrobiology. Birbal Sahni Institute of Paleosciences, a bunch of my colleagues there are using paleontological approaches to try and understand ancient minimal living systems to sort of, they use a top-down approach to understand origins of life. And here I've also mentioned PRL, where some of the interesting work is going on.
So I don't know whether you have heard of Professor Ponnam Peruma. So
Cyril
yes,
Ponnam.
Cyril Ponnam Peruma. When I was a graduate student, which was like about 100 years ago, so I used to visit Bangalore quite often, and he was a perennial fixture there.
Yeah.
And he used to give these very nice talks on astrobiology.
Yeah, wonderful biochemist. Yes, yes, I've read some of his papers. Very inspiring.
Yes, so he was from Sri Lanka.
Yeah.
But he worked in the US.
I see.
And,
Very nice, very nice. Thank you.
all
So
right,
much.
thank you very much, Prachi. Now,
Okay, thank
it's
you.
up to you now, I may have to leave in a little while, but
Okay, yeah,
Thank you for being here. Thank you.
thank you. Over to you, Gayatri, for questions.
Yeah. Thank you, Dr. Sudha, for such a wonderful talk. Like, we have a questions on Zoom as well as on Facebook. So I will read out. So I don't know the name because it is showing the device here. The chemical or biochemical processes you have described, are they time invariant? I mean, the student is saying, do they need a long geological time to take place? Or if not, how do you model those time invariant reactions in lab?
Oh, yeah. So this is a very, very relevant question, right? See, all the events that I talked to you about are thought to have happened over very, very, very large time scales, several millions to billions of years ago, right? You and I are never going to exist on those time scales. So it's very important to sort of think about how you could not just encapsulate some of those processes in a more pragmatic manner, but if you do understand how they can happen in shorter time scales, is there any way to take that information and feed it to, say, for example, a computer program that allows us to model, for example, what might have happened over much longer time scales? One of the important limiting factors here is our capacity to be able to model such very, 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 very huge timeline processes. We still haven't been able to get to that point. But I am hopeful that with this crazy explosion in artificial intelligence and related softwares, maybe we might not be too far away from that day when you can probably model this whole process from start to finish and learn a whole bunch of things from it and refine your experiments in a manner which is, again, more pragmatic than when you started out, and iterate this process till you basically try and fit all the remaining pieces of the puzzle. This is not going to happen in our lifetimes. It's going to take a lot of work over a few lifetimes, but it nevertheless is very, very promising, right? Please don't forget, the field of prebiotic chemistry came into being in 1953. And already by 2023, we are able to talk about all these different things. And if anything, science and the tools of science are exponentially growing. So maybe we are much closer to, for example, using the fundamental building blocks, forming the minimal living system in the lab in a test tube. Is it 5 years down the line, 10 years down the line, 25 years down the line? I cannot really say, but it's much closer to now than being much farther away. So I hope that's indirectly answered your question.
Yeah, thank you so much. There is another question, Prabir is asking, can you please briefly tell the process of development of DNA from RNA, why it took so much time, like 1 billion years?
So we do not know if it was 1 billion years. We don't know that for sure, right? Right. But why did you go? Maybe the better way to phrase this question is, why do we think we went from a probably fully RNA world space to a space where you have both DNA and RNA? So one of the very important things that I touched upon, albeit ultra briefly, is that in the presence of prebiotically relevant co-solutes, your rates of replication of RNA-like molecules and the information therein might have been more mutation prone. Yeah. And this is because RNA intrinsically is prone to higher mutation, even in modern cells. And back then, the kind of mutation rates that happen comes from a slightly different, comes because of a slightly different problem, which is you did not have enzymes. So there was no error correction mechanism. And you have what is called wobble pairing, right? So you're using a template, you're using a primer, and you're hoping that completely by a chemistry-driven process, you're going to have the right Watson and Crick base pair come in, bind to the template base, and stitch the primer and start extending it. However, please don't forget, as much as G base pairs with C, G also loves to hang out with U. It's a very prominent base pair, even in biology. Despite all these enzymes, you see this wobble pair very prominently. So even intrinsically simplistic chemical mechanisms might have led to a lot of these mutations, quote unquote mutations, to have been plausible on the early Earth, which was all not bad. Initially, it was important because in this RNA world space, if you want to sample all possible molecules to finally find some molecules which have a catalytic function, the timescales we're talking about, even though we had enough time, let's say over billions of years for this to have happened, we are still talking about a vast number of molecules, right? If you take, say, for example, a 25-mer RNA, and just based on modern bases, which is 4 bases, the total number of all possible 25-mer RNAs goes to what numbers? 4 raised to 25 molecules. That is a huge sequence space. So if you want to sample all of that sequence space, higher mutation rates would have been good, because then it allows you to sample all possible sequences very quickly. So in that sense, early on, RNA world would have been advantageous. But once this RNA world molecule was explored and interesting functional molecules came about, like a catalytic RNA molecule, which could stitch amino acids to form peptides, so on and so forth, once you started having encoding of the information and transmission using proteins, it would have been much more useful to have a polymer which is less prone to mutation. And DNA is less prone to mutation. And DNA also is capable of forming double-stranded structures much more readily, which allow for information transfer in a very, very quick manner. So because of this, there's a notion that you transition from an RNA to a DNA world eventually, wherein in this world, DNA plays the main role of encoding information. RNA still encodes information in some viruses like your SARS-CoV-2 virus, but it went on to do a lot of other functions which it's awesome at, which DNA cannot do. DNA can encode information, and that's about it. Encode information, keep the information safe.
Okay. Yeah, thank you. So there is a question by Dibyo Majumdar. It is like, did these processes take place randomly in the primitive Earth, or did some factors guided these processes?
Guided. Now that's a very tricky term. Yes, maybe some physicochemical events guided it. What do I mean by that, right? Or some substrates guided it. What do I mean by that? This formation of RNA polymers from RNA monomers, if you think about it, is a very uphill process. I talked to you about that. And I spoke about one hypothetical pathway by which this can happen, wherein lipids are involved. Now, let's say you don't have lipids, but you're looking at a scenario where, just like your volcanic geothermal system, what is constraining the water in that space? It's the minerals that surround it. It's the rocky structures that constrain the water in that pool, right? A lot of these rocky structures, stones, are essentially minerals, which provide extremely interesting surfaces for facilitating some of these reactions much more readily. because now your RNA monomers are not trying to sort of meet each other in a three-dimensional crazy Brownian motion-driven space, but they are now more constrained by a mineral surface where they can be in much closer proximity and undergo these uphill reactions. So in that sense, it's thought that mineral-guided catalysis was a very important step in forming higher polymers, longer polymers, basically going from the monomeric space to the polymeric space. So in that sense, yes, there were certain naturally occurring geological and geochemical events that are thought to have guided, in your words, or shaped, in my words, that are thought to have shaped several of these prebiotic processes. I hope that answered that question.
Yeah, thank you so much. There is a question by Amol. He is asking, does there are evidence of life other than carbon waste?
So see, carbon is a mind-blowingly awesome atom. It can do certain things so much more readily than, for example, its very close cousin silicon. Silicon forms compounds which are very, 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 very stable. So you form them in the forward direction, it's extremely hard to go back in the backward direction. With carbon-based compounds, you can actually make and break bonds much more readily, which is much more interesting when you think of, especially when you're thinking about processes where you had to form several different kinds of molecules from certain small set of ingredients. So for that reason, I cannot think of another atom that can come close to carbon in terms of doing all the stuff that carbon can do. Hence, if I was looking for life forms in some exoplanet, I will still focus on carbon because the chemistry that can be driven by carbon is absolutely magnificent. But that doesn't mean we close our eyes off, right? We are open. We keep our eyes out, and if there's something that's outside of the ordinary, we still try to understand what it is.
Thank you so much. So actually, one of the participants, Neil Gaikwad, he's asking that he's interested in this subject. So how one can get in touch with you? Like,
the best way to get in touch with me is by email. However, please understand that there is a physical limitation of how many people I can host in the lab. So some of the things that you can do much more readily is a reading project, because there's a whole context to this field that is not taught to you in schools. You probably hear about the Stanley Miller experiment, the Stanley Miller and Harold Urey Miller experiment. You probably hear about the Harold Operin and Haldane hypothesis, not Harold, I'm sorry. But you don't hear about much other very important geological context, planetary context, organic context, geochemical context, yeah? So there's a ton of reading to do to even set the stage for better understanding of some of the projects that we do in the lab. So maybe a reading project is possible. However, please remember, there are a whole bunch of these other institutes where there are people doing a lot of interesting things. So depending upon your favorite area of sciences, you have more people in the country to hit up and check if they can host you. But like I said, we are generally very open. It's just that we always deal with a whole bunch of requests. So you might be in some aap Qatar mein ho kind of a response, right? So please don't mind that.
Yeah. Thank you so much. There is a last question from, I can take, this is from Yogesh Kane. He's asking, there is evidence of relatively sudden diversification and proliferation of advanced and complex species during the Cambrian era, that is from simpler to unicellular. Are there any key biochemical changes at biopolymer level that could have triggered it?
You know, this is something that we still do not understand very systematically. What led to this explosion in complexity, right? Like you very correctly pointed out. One of the things that I didn't sort of mention. Yeah, let me see. Yeah. About right now, the oldest evidence is close to 3.85 billion years ago. There's enough reason to believe that the first forms of life, the prokaryotes, probably populated the planet almost by 3.85 billion years ago. These are cyanobacteria-like prokaryotic systems. Here you're seeing what are called stromatolites. These are pillow-like rock formations that you see even today in certain water bodies. And in this case, you're seeing it. This is a water body from Perth, Australia. These are dead stromatolites, but you also see living stromatolites. They are essentially a byproduct of interaction between biology and geology. And if you actually study these closely, what you will realize and date these systems, you'll see that they're really, really, really ancient. So it's thought that, see, if you put all of that timeline, that billions of years of timeline on a clock, like I'm showing here, some of the oldest rocks are from 4 billion years ago. Some of the oldest definite fossils that we see of prokaryotes, like what we see, what we sort of have discovered in these stromatolites, or even certain hot spring deposits, date back to about 3.85 billion years ago. So you're still around. I mean, you've all the way come to noon, and you're still in the prokaryotic space. From there, it's thought to have taken another couple of another billion years at least, or 1.5 billion years at least, before eukaryotes or our ancestors came about. Yeah, here is an example, kind of a example to give you a sense of one of our ancestral eukaryotic species. All of these entities are thought to have come about only about 2 billion years ago. Now, Cambrian explosion, if I'm not wrong, would have happened about how many million years ago? About, I think, 630 million years ago, right? During this period, there's been a whole bunch of evolutionary processes that involved complexification of the eukaryotes to other kinds of more complex entities. One of the very important things, one of the very important biochemical necessities for increasing the complexity of an organism is for it to be able to encode more information, more useful information, which will essentially allow for the formation of a whole bunch of molecules that can do a whole bunch of higher order functions. That is one understanding for sure. But what are the environmental parameters and what are the physicochemical events that happened between this time scale that led to an explosion in complexity from eukaryotic systems to simple multicellular systems to Cambrian space, where you had organisms that looked like, I'm sorry, I don't have that slide with me, but all kinds of crustacean looking complex organisms that populated the water. How did you go from multicellular simple species like these, where you have these choanoflagellate rosette colony, to way more complex, large crustacean fish-like looking creatures? We still do not know. Surely you needed an increase in capacity for encoding much larger amounts of information, but what set the stage for this? Still unclear, at least I'm not aware.
Okay, thank you, Dr. Sudha. Thank you so much for your time. This was a really interesting talk. And we also want to inform participants that we have shared a feedback form link in the chat box. Please let us know your suggestions, like what kind of topic you would like to cover as a part of these kind of series. And thank you, Dr. Sudha, once again. And we also request participants to turn on their camera so that we can take a screenshot. And this talk will also get published on our YouTube channel. So please, in the next week, you can also find the recording of the same talk. Yeah, thank you so much. We request participants to turn on their cameras.
Yeah, thank you all for joining. It's a shame we can't do this in person. Randeep, hi. Man, you joined. You shouldn't have joined. I hope I lived up to your expectations. Good to see you, see some of you that have, yeah.
Yeah, thank you so much.
Yeah.
And Dr. Sudha will look forward for the course in the astrobiology. We'll connect you separately for the same.
Sure, sure.
Thank you so
much. In the next couple of months, but surely in the immediate future.
Yes.
Yeah. Okay,
sure,
thank
sure.
you all.
Thank you so much. Thank you. Thanks. Bye.
Thank you. Bye.